Now, the advantage of this approach would be, if we can do it, this is a harder experiment than the previous one. It involves getting a protein, an amylase, into the brain. But the thing is, if we can get it in only one time, then it will digest what's been accumulated for 50 years or 60 years, and that's it. We don't need to get it in again. The patient can then form new polyglucosans for the next 50 years of their life till they're 110 years old, and we won't need to intervene till then. So this would be a great thing if we can get amylase into the brain, into neurons, one shot. Not easy, but a possibility. Now, in order to see if this would actually work, we can try an experiment at the genetic level, which I'm going to describe. Again, it involves breeding of mice. So we would take Orhan's mouse, the adult polyglucosan body disease mouse, and we would breed it with a mouse that we have engineered in our lab, which produces amylase in the brain in neurons. So when we breed these mice, they will have adult polyglucosan body disease, but their brain cells will produce amylase in the neurons. And hopefully, we will see whether the amylase will digest the polyglucosans and eliminate the disease. So this is the second experiment. It involves a breeding of adult polyglucosan body mice with our amylase mice. In summary, we're proposing two experiments. Both of them are breeding experiments. One will breed the mice with the amylase mice, and one will breed them with the PTG knockout mice, follow them for 6 months, and see if we can get rid of the polyglucosan bodies. Here I wrote a summary of the two experiments. Experiment 1 is the adult polyglucosan body mouse bred with the PTG mouse. What will this achieve? It will prove whether downregulating GS actually stops and prevents APBD the way it does Lafora disease. We know this in Lafora disease. We need to know it in this disease. This will focus subsequent work on finding a small molecule that interferes with and slows glycogen synthase activity. And as I said, polyglucosan accumulation is probably, in my estimation, a net accumulation. That is the difference between their production and elimination. Potentially, stopping production could result in natural gradual elimination and cure. So this experiment will open the way for a treatment to stop the disease, but potentially also to cure the disease. The second experiment, which would go on simultaneously, is breeding the branching enzyme mice with the amylase mice. This will prove whether introducing amylase into neurons clears polyglucosans and cures APBD. This will focus subsequent work on finding a way to introduce amylase into the brain only once. Single introduction should clear polyglucosans accumulated for 50 years and thus allow freedom from disease for the next 50 years. So these are the two experiments that we proposed and that we were asking for funding for. They can go simultaneously, and there's a lot of money saved by doing this in one shot together at the same time. It's only possible because the branching enzyme mouse is available now through the work of Dr. Zachman, Di Mauro, and their group. This is the end of my presentation.
Thank you very much. I just want to also send a special thank you to Gregory Weiss, who is listening. He's not on the computer. I'm phoning him now to find out if he has any questions, and I want to thank Dr. Minassian for taking the time to prepare the presentation and do a couple practice sessions and come on with us today. If you have any questions, please press the button right to the left of your name and raise your hand, and you'll be unmuted. And you can...
That is Minassian presentation.
Oh, he can hear.
And yes, and all that I expected. Dr. Minassian, thank you very much. Now we need you to prove, as I said in the past, that the two hypotheses, at least one of the hypotheses, can really work. We need to bring it to reality.
Yeah.
Now, question to you really is, going forward, once you prove this on the two mice, how do we really back to human testing? What steps are needed once you find the small molecule to approve it, to get it to human testing? What is next?
Okay. So, on the LaFora side, we are already working, and this is our main focus in the LaFora work, to try to find the small molecule that will inhibit glycogen synthase. I've had already conversations with Orr, and he has given me great suggestions, and I've searched myself also. There are potential already existing medications that we can use. But there are all sorts of issues and problems with each of the medications. Most likely we will have to find a new medication, and to do that we will need to screen libraries of small molecules to find one that actually inhibits the glycogen synthase. So we're setting up an automated system to test many small molecules that might inhibit glycogen synthase. What this means is the assay that I described, that molecular pathway that I showed you, we are trying to automate it so that we can screen thousands upon thousands of small molecules which are already available. Some of these, several thousand of them, are FDA approved molecules. These would be medications used for any number of different diseases. If we are lucky and one of them inhibits glycogen synthase, then we have a medication that we can immediately try on patients without the need to get any type of FDA approval because it would be already FDA approved. Then there are thousands of small molecules that are natural compounds that are available. And these also will need very, very little, if any, regulatory intervention because they are stuff that are consumed naturally. So if we're lucky and the chemical that we find is in one or the other of these groups, let me just go back to that picture for anyone who's kind of lost track. So if the small molecule, which I depicted as a pill here, is something that is either a natural compound or FDA-approved chemical, then we can use it right away or almost right away. If it is not, then we have to go through animal studies and so on, and that's longer, obviously. But, I don't know. It depends on the molecules. We may get permission to try it on, our most difficult patients and so on. Those aspects I have not exactly thought through because we're not there yet, but these are things we need to think about and start planning for as we move forward. So we are currently doing the work in LaFora that will benefit the APBD, the search for the glycogen synthase inhibitor. Now, of course, the more money I have, the more I can do. We're doing it on, budget from, again, patient groups, from LaFora patients and so on, and other sources. But I think for the APBD, for the moment, I'm asking the association to fund the mouse part so that we know in APBD what we already know in LaFora disease. Without that knowledge, it's still theory. So we need the proof. That's why we need to do the mouse studies. That's regarding the small molecule. Regarding the amylase one, it's a lot of work and very difficult to get the amylase into the brain. So investing the large amounts of money and effort to try to do that is kind of silly if we don't know if it will work in the first place. So we need to do the mouse experiment as soon as possible to know as soon as possible whether it will work. And then we can plan to try to see if we can find a way to get the amylase into the brain. That's the end of my answer.
Emil has a question. I'm going to put him on now. Emil, you're on. Emil, are you there? No, he's not there. Anyone else have questions? And did you speak at all about the timeline that you were thinking of for these two?
Yeah, the mouse experiments, unlike most things in research, which is usually very hard to give a timeline, these experiments we can actually say exactly how long it will take, unless some dramatic, unexpected problem happens. The reason we can do that is because Orhan has already studied the GBE mouse very nicely, and we know from his work, from his publication, precisely at what times the polyglucosans form, and so we plan the experiment to last about 6 months, and so I'm giving leeway, a few months before to get the mouse breedings and so on, and then a few months after to study the pathology and etc. So it's for the first time relatively easy to say how long this will take. In 1 year from when we start breeding, we will have the answers. So if we meet 1 year from now, we will know the answer to these questions from these two experiments.
Leo has a question. Hi, Leo.
No, I was being called, and I just wanted to say hello and tell you that your webinar has been prepared very wonderfully, and kol hakavod. I congratulate you on your wonderful work here, and it's extremely informative, and again, we're very, very well prepared. So I thank you.
Thank you very much.
We're recording it, and we're going to try to have it available.
I look forward to seeing it a second time because I couldn't absorb everything, and all the information being presented is exactly what I wanted to hear.
Thank you.
So thank you.
Hey, anyone else with any questions?
Minassian, I'm back here. May I?
Yes, please.
Talking now, this PPG mouse that you already have, aren't you looking at or using a small molecule to suppress the glycogen synthesis? What are you using in that now?
Yes, we are. This is what we're doing, Mr. White. We're searching for, we're getting set up to be able to search for the small molecule that would inhibit the glycogen synthesis. All these automated experiments that I tried to explain before, that's what we're trying to get set up. Unless we automate it, we cannot screen thousands and thousands of molecules, so we need to do that.
Okay, so we all together need, number one, to thank you, number two, to wish you and all of us together lots of luck. Thank you again.
Okay, thank you.
Very much. That ends the webinar.